My name is Asaf Singer, and I'm honored to present in front of you today my research entitled Enhancing Inflammation Targeting Using Tunable Leukocytes-based Biomimetic Nanoparticles. Biomimicry, literally imitation of the living, aims to take inspiration from natural selection solutions adopted by nature and translate the principles to human engineering. The biomimicry approach aims to favor choices tested by nature, which had millions of years to understand what works best and what doesn't. Designs following biometrics will ultimately allow human productions to be more efficient, resilient, and sustainable. Biomimetic nanoparticles aim to effectively emulate the behavior of either cells or exosomes. Leukocyte-based biomimetic nanoparticles, for instance, incorporate cell membrane proteins to transfer the natural tropism of leukocytes to the final delivery platform. Why do we want to mimic leukocytes? During inflammation caused by tissue injury or chemical agent or any pathogenic microorganism, our capillaries getting wider. This is leading to increasing capillary permeability and then white blood cells are attracted. So we have asked ourselves if we can mimic the way white blood cells target inflammation using leukocytes biomimetic nanoparticles. To answer this question, we have to look closely on the leukocyte association mechanism to inflamed endothelia. During inflammation, lymphocyte function-associated antigen 1, LFA1, permits leukocytes to transiently bind to intracellular adhesion molecule 1, ICAM1, which is overexpressed in activated endothelia. After understanding this specific mechanism, we have created a kind of blueprint for this fabrication of these specific biomimetic nanoparticles. First, we cultivate leukocytes in vitro. Then, we extract their membrane proteins using a specific procedure, and using a microfluidic approach, we fabricate our leukosomes. The next steps are mainly physiochemical and biological characterization of our biomimetic nanoparticles. At the end, we provide the medicine. I really like to look on our biomimetic nanoparticles as Lego blocks. We decide what will be the membrane proteins on our nanoparticles with respect to the cell source and the purpose of the targeting. Then we decide what will be the nanoparticle's backbone based on the theranostic cargo that we want to deliver, and then we just fabricate it. From an engineer point of view, I've asked myself, will increasing specific proteins content on leukosomes result in a corresponding increase in the inflamed cells uptake using two different in vivo models? So what we have done. So as I mentioned before, we cultivated the leukocytes, extracted the membrane proteins, but this time we created 4 groups of nanoparticles. The first one is liposomes, which is a lipid nanoparticles without any membrane proteins. Then we created the leuko 1 to 100, which reflect the lipid to protein ratio on the particle. So leuko 1 to 100 will be the particle with the lower concentration of proteins integrated in the membrane, while leuko 1 to 20 will be the one with the higher concentration of protein. Leuko 1 to 40 is an intermediate, is a particle between these two groups. Then at tile 4, we characterize these particles, and then we tested them in vitro, followed by in vivo testing in 2 different in vivo models. One, gram-negative bacterial infection, which we induced by lipopolysaccharide, and triple-negative breast cancer. The physiochemical effect of lipids to protein ratio on leucosome was assessed using dynamic light scattering and cryotransmitting electron microscopy. The only physiochemical effect that was noticed is the decrease in the zeta potential with respect to the amount of proteins integrated to the membrane. Higher protein content led to more negative zeta potential, as we also reported in our other papers. Then, using Western blot for specific leukocytes markers such as CD11b, CD18, CD45, and CD47, we verify the increase of the protein in our nanoparticles group, as you can see in tile G in front of you. We also assessed the stability of these nanoparticles for 21 days at 4 Celsius degrees. We did not assess any statistical significant change between the different groups. So what I'm trying to claim is by adding more proteins, we didn't change the stability of the nanoparticle. Then we assessed the enhanced targeting using high throughput flow cytometry and confocal microscopy. As you can see in front of you, the leucosome 1 to 20 group, tile 4, and the red bar on tile B target inflamed endothelia better than the other groups. You can see the increase. We have used intravital microscopy to observe the different nanoparticle circulation patterns in live mice at high resolution. And then we noticed higher circulation time for our leukosomes on the right compared to the liposome circulation. In green, you can see the blood vessels. Red is either liposomes on the left or leukosomes on the right. Finally, in vivo imaging system was also used to assess the enhanced leucosome targeting using 2 different in vivo models. The first one is a lipopolysaccharide-induced local inflammation in the right ear of the mice. As you can see in front of you guys, the 1 to 20 groups, the one with the higher protein content target the ear better than the other groups. The second model that we have used is triple-negative breast cancer. And the same effect was noticed also here. The 1 to 20 group target the tumor vasculature better than the other group as well. Finally, we have demonstrated that by increasing the membrane proteins in our biomimetic nanoparticles does not correlate to increasing toxicity, as you can see in front of you from the immunohistochemistry analysis on the filtering organs compared to a control organ. 4 takeaways. Drug delivery strategies can follow rules and modalities of cell-to-cell communication. Second, biomimicry is a designing principle to engineer nanoparticles inspired to cellular components. Third, leukosomes resemble the proteolipid composition and preserve the function of leukocytes. And 4, leukosomes target inflamed endothelium. I want to thank you all for listening. Thanks.